Welcome to this lecture, and I would like to introduce Susana Cristobal, who's a professor in biomedical research at Linköpings University. She is studying nanoparticles, and she will speak about risks and benefits with nanoparticles in our life, in medicine and in other situations, if I have understood correctly, right?
Right. Thanks a lot. Thanks, and I'm really happy to see you here to talk and to discuss about this topic. First of all, before going ahead with the presentation, I really want to thank to my team. Science doesn't go really too solid without the help of your team, and my team is a group of very talented young scientists that helped me a lot to go from the vision to the really solid results. So thank you all. And now we go into our topic. We are going to talk about nanoparticles and nanoparticles that are around us and why this size matters. Today we see that nanoparticles, that is defined as those components that do have one dimension between 1 to 100 nanometers, are surrounding many of our consumables, coming from sunscreens, painting in our houses, textile, food packaging. More and more of our daily consumables do contain actually some small nanoparticles into there. But, and this is very important in the context of the nanotechnology that is going to introduce even more of those in the future in areas like nanomedicine that hopefully could go to target to specific cells where the drug will deliver and will help to our and some aspect of our lives. But nanoparticles are not something new actually, has been in our life for a very, very long time, most likely since we start working with fire and dust were spread around. So one way of having a look to nanoparticles around us, I want to present in two different aspects, from the new perspective and from the old perspective. Why we can see the nanoparticles today? We can see because we have technology that allow us to see, such as transmission electron microscopy. And I put in the top of this slide some examples of how we can see these nanoparticles today. Titanium dioxide nanoparticles or other more sophisticated nanoparticles, supramolecular base. But if we try to think about these nanoparticles that's having around us all the time, I want to go through this example of the silver spoon that our mother or grandmother do have at home. If we take a silver spoon of those ones and we put in a grid of one of electron microscopy, we let it there for many hours, maybe a day or so, we remove it, we have a look what's happened, and what we could see is that we have nanoparticles over there. This is a very simple, elegant experiment that tells us we have been surrounded by nanoparticles in our life. So, what type of nanoparticles we can consider? We can consider those that are naturally occurring nanoparticles in nature, from volcanoes, from spread from the water, from the sea, from dust, and those man-made nanoparticles. But even when we consider man-made nanoparticles, there is also different type of those nanoparticles. We can consider those that happen accidentally. Those have a variable size, they are heterogeneous, those happen maybe through friction, they could happen in different shapes and different elements. And maybe the ones that are our main concern today are those that we call engineered nanoparticles. They are specific made with a well definition of physicochemical properties with a particular purpose of having a specific functionality. But thinking about when we were exposed to engineered nanoparticles, actually is not that recent as we thought. We have evidence that Roman, Chinese, Egyptian, they were already prepared engineered nanoparticles from golden. And actually some of the methods that they utilize are not that far from many methods of synthesis that we use today. So this is not a story really new. It's a story that has been around for a long time, but the potentiality of nanotechnology come to a context where we need to concern about the exposure and the possible risk, because the production increase and the risk of exposure increase as well. When we think about the relative sizes, to give a perspective of what we are talking about and why size matters, we can have this kind of a scale where we can see a bulk material. A bulk material could be the same kind of chemical component than a nanoparticle, but different size. And a bulk material could be between 100 nanometers and 10 micrometers. Then we have the definition of nanoparticles between 1 nanometer and 100 nanometers. But we can see also at this end that there are nanoparticles that ultra small, maybe 1 to 3 nanometers. Those are really very close to molecules, molecules or atomic level of 0.1 nanometers. If we want to think about our everyday world, where a meter is, let's say, the standard of size, then we will be in the context of 10 to the 10 nanometers. This is the perspective of the sizes of nanoparticle in the perspective of our daily life. One thing that I want you to have a look and consider to understand these aspects regarding to the physical properties of nanoparticles and the comparison between macro scale and nano scale. Where are located the majority of the atoms when we are thinking in a macro scale? The majority are actually inside the particle compared to the surface. If we think about the nanoscale, actually are divided almost equally between the surface and the inside. But if we modify the size to something smaller, we have an effect. This effect is smaller in a macro scale. Still, we have the majority inside compared to the surface. But if we go to the nano, if you can see my drawing over there, the surface is actually almost everything. And this actually happens if we think about nanoparticles 1 to 3 nanometers. This is why the surface is almost everything, and the surface increase enormous, and also enormous this reaction capability. And this is one of the aspects that are interesting for the properties of these nanoparticles. If we go to some aspects like optical properties, I take an example of gold nanoparticles and gold as a component. If we take bulk gold molecules, appears yellow, everybody knows. And if we go to nanosite gold, actually appear red. And the reason is because the electrons from a small particle cannot move freely compared to the bulk material. These movement constraints compress the particle reactivity with light and therefore the color. Another example, zinc oxide. If we go to the bulk, it's actually white because scatter light, the visible light. But if we go to the nano size that actually is used as blocking UV light, in this case it's clear because visible light cannot be scattered. So we can see here the difference in these properties, in these optical properties, how this compromise those aspects. Let's take another type of nanoparticle made of carbon, like carbon nanotubes, that are very interesting regarding to optical properties. Carbons are really long cylinders of carbon, and they have very, very fascinating properties. Could be 100 times stronger than steel, still very flexible, and still unique properties regarding to electricity. And these properties are related to this structure that also could be modulated in different aspects. They could be formed just one single well, they could be formed a multi-well, and these properties, these changes in diameter, these changes in structure, is what define this structural behavior. And if it changes the conductivity to semiconductivity, and this is the play aspect that material science utilize in order to make new engineered nanoparticle specific properties. So what we can see now from this properties is that size matters. Size matters when it comes to properties in nanoparticles. And size matter and make that application has been spread enormously. In this particular slide, I have taken from commercial scale production of inorganic nanoparticles. And in the center, we can see those areas where the manufacture is more potent. And those are textiles, biomedical, healthcare, food and agriculture, industrial areas, electronics enormously as well, and in the environment, in renewable energies, are also areas where nanoparticles has played an important role. So we want to see also not only the aspect related to the size and the aspect related to the properties, but also how this size matters when it gets into interaction with a cell, and what could happen, and what could be the risk, and what is this physical property connected to the possible risk exposure. And I think that this should be understood in this particular aspect of a balance. We are trying to understand the balance between the benefits that those new formulations could give to our daily life and the possible risk that also could be involved. And we as a scientists have this role to offer a platform to evaluate this possible risk, and also maybe the platform to offer how to evaluate the new formulation of nanoparticles in order to make a design that fulfill specific properties that are desired, but at the same time fulfill the specific needs of lack of risk or small risk for us consumables of components that do contain nanoparticles. And this is what we are doing in our daily life in the university, in my group, working with these questions. So what are the possible risks and how we can understand these possible risks of exposure? I'm going to present this with three different aspects. The first aspect, I'm going to focus in the organismal perspective. Let's say we're thinking about a human being, and a human being has been exposed to accidental release of nanoparticles. In this case, we have possible routes of internalization. These possible routes are most likely inhalation, and they will go through the inhalation and they will deposit in some cells into these channels in the respiratory tract. They could go also through circulation and be absorbed through the vessels and into the circulatory system, could be circulated through other organs, liver, kidney, heart, could go through the blood-brain barrier as well, and nervous system. Other possible areas of exposure could be maybe skin or damage into the skin, or maybe through some particular orthopedic implants. What we see from the literature is that from this organismal perspective, our risks in different organs could be seen from this kind of degree. Maybe it's higher for liver than kidney, than spleen, than lung, brain, and heart. And finally, maybe we can have a look to the last or the area of this slide when I try to put what could be the relation between this exposure and the possible adverse pathway for health. And this is a kind of extrapolation of if these nanoparticles end up in the respiratory tract, there is a risk in some context of inflammation, asthma, and other type of diseases. If it's related with circulatory system, this could be related with cardiovascular diseases. But we shouldn't be very concerned about this final risk. We need to understand the aspect of the, before going into the understanding of the organismal perspective, we need to go to the understanding of the cellular perspective. The first thing is that we need to make a correlation between all of these physico-chemical properties of nanoparticles and how these affect to the cellular impact. I have made a drawing of a cell and the main organelle components here. And I want to present it that the nanoparticles most likely will be interact with the cell in the membrane and will be invaginate into a kind of vesicle, and this vesicle will end up in the cell. They could end up with ROS production, ion leaking, and things that will involve possible risk related to oxidative stress, lipid peroxidation. They could end up also in the nucleus. Ultra small nanoparticles are so small they really enter in the nucleus and could affect to genotoxicity. There are aspects related with mitochondria damage that also can be observed, and also apoptosis. There are also very small nanoparticles that actually could get into the cell without a vesicular transport, rather like small atoms or small nanoparticles, and those also could affect in the same aspects. So this is the overview of the possible risks at the cell perspective that could observe. And I want to have to the third aspect. So we have see a little bit possible risks regarding to the organelle perspective, possible risks regarding to the cell perspective, and now we want to take your attention in an aspect of the classical toxicology. Classical toxicology uses this kind of curves of dose response to try to understand the effects. And in the upper part of this slide, what I have put it is a dose response, let's say, of 3 different pollutants or components that we want to evaluate, completely different from the chemistry perspective. And each of them do have a different dose response. We can test it, we can evaluate, we can understand what's happened. Why nanoparticles become more complex when it comes to toxicity? Become more complex because we are in this scenario, and I try to explain what I tried to draw in there. One single component, let's say, we work in our lab with titanium dioxide, with this chemical composition, still can be produced in many different sizes, from 1 to 3, 20, 30, up to 100. Still can produce also with different coverage, still can produce with different arms for specific targeting. So what's happened? We have one component, but still we cannot study from this perspective, increase in concentration, what is the response, because each of them is a different one and could respond differently. So we get a kind of completely different perspective that is much more difficult to handle in a case-by-case scenario. That is the classical toxicology approach. This is why we think that there are some key questions that we would like to understand and will help us to understand this aspect of nanotoxicology or NANOSAFE and make this balance. And I think these questions are, what are the difference between the nano and the bulk material? What happened to the nanoparticle when is into the body? Because once we have inhalate or get in contact with the skin and end up into the organism, it's going through different medias, different environments, and the nanoparticle will change, will evolve, will be surrounded by different molecules, will be affected by different molecules. What's happened to the body if we internalize the nanoparticles? And finally, but maybe a little bit out of the context of this talk, what happened to the nanoparticles in the environment? That is very complex regarding to the study of the nanoparticles alone because of the different components that we have there, but still is a kind of basin of this release and also a way to end up also into humans as well. So my final question is also related to how can we utilize this type of methodologies that we apply in the lab, not only to understand toxicity and safety, but also to understand how design nanoparticles with specific properties and how to make it more safe. So our vision is that we want to utilize this, our techniques as a shortcut because we cannot go case by case. And in order to do that, we work in a platform that try to address these questions. We try to address which nanoparticle are going to be targeted to a specific cell, which one has the possibility to be uptaked, what could be the possible risk on nanotoxicity, and what is the impact and risk of these nanoparticles. If we're starting from the target perspective, I want to explain you with this kind of drawing. I put here a doll, and the doll here represents a nanoparticle. When we produce a nanoparticle is pristine, it's like naked, but it's just there when we produce it. Once we are dissolving in a substance and get into a media, there's not any longer by itself. It's surrounded by many other molecules. We have these molecules in our body fluids. So nanoparticle is going to cover by these molecules. These molecules most of the time are proteins and forming a protein corona. So what will happen, under my perspective, is that depends on what is the molecules there, the nanoparticle will have the opportunity to be surrounded by different proteins. And this is specific for each nanoparticle and depends of the physicochemical properties. But by studying this nanoparticle corona, we can actually have a shortcut of prediction that I was presented. And this is because if you see this doll that in the upper part is dressed for going to a party, in the bottom part is dressed for going to the mountains. Actually, this cover, this dress, is giving a lot of information. Because if you are dressed for going to the mountains, your risk is maybe to have an accident. And if you are dressed for going to a party, your risk is maybe to get drunk. So by knowing this kind of coverage, we really have a lot of information for prediction. And this is what we do in our lab. So in a few slides, I will present little pieces of our research to try to explain this idea. And this is the first, that here is the way how we do that. We take our nanoparticles and we incubate it with this media. The media is the first media that the nanoparticle meet. And we let it form the nanoparticle corona. Then by utilize analytical chemistry, mass spectrometry, and quantitative proteomics, we are able to extract those proteins that form the corona, identify those proteins, and quantify the relative abundance of those proteins there. And this is what we do in our lab for many different nanoparticles and concentration. And I was putting here a piece of this data that I hope is easy to understand. In the first part of this graph, I have the total abundance of protein and the distribution of the proteins in this first media that I use to incubate my nanoparticles. And in the others, I have the distribution of proteins in the corona of different nanoparticles. And what I try to illustrate here is that it changed completely the distribution of these proteins. And by knowing this change and by knowing the distribution of which proteins are, which biochemical pathways are involved, I can understand the possible risk of this nanoparticle when interact with the cell. And I could understand also which is possible if this formulation is likely to be targeted to a specific cell. Because when we define some nanoparticles maybe made for, with the goal of drug design, we also maybe need to have some specific arms or specific elements that need to be seen by the cell for the targeting. By utilizing this methodology, I will be able to show and to get information if these elements are really visible for the cell. And therefore, this formulation is likely to reach the target cell and is likely to make the specific function. If I go to the aspect related to the target and to the uptake by the cell and therefore the possible risk of the cell. By utilizing this methodology, also we can not only understand this nanoparticle corona, but also understand the evolution of this nanoparticle corona after uptaking by the cell. Because what I'm telling at the beginning is this nanoparticle is not pristine, is always covered by different molecules, and this is evolving and changing in this process into the cell. So what I do now is to take this corona and then incubate with the cell content. And then I study this evolution of the corona. And this I can do in different aspects, depends on the design, if I'm thinking about something that goes through different steps and is evolving and changing. But in this particular case that I present here, I use cell media for some cells and cell contact from identical cells and study this evolution. I'm going to put just one piece of a slide to show you this data from endothelial cell. And as in my previous slide, I present here also the total content, the abundance of the protein from the cell, and then the composition of the corona and the change in this corona that is so drastically from the corona that we observe at the beginning just in the media. And this is also very different depending on the size of the nanoparticle that we utilize. We have tests with the same chemical composition and different concentration and sizes. And we can, from this information, we can go to prediction, as I told you before. And how we go from that? We go from aspects related to, we can identify what are the key events, what are the nodes of this connection between the protein's abundance. And these key events could be related with the risk that I was presented at the beginning. I hope I could convince you that utilizing these methods could help us to understand the capability of some nanoparticle to be targeted to a cell, and also to predict to some aspects of the possible risks of the nanoparticles when it is in the cell. I would like now to discuss a little bit more the nanotoxicology aspect and how we can address this aspect. And this aspect can, when we think of nanotoxicology and the methodology that we want to use it, there are one aspect that is important, and this aspect is related to characterization of the nanoparticle. Because, as I told you before, there is a myriad of different nanoparticles that we produce nowadays, and with different physicochemical properties. It's essential to understand these physicochemical properties in order to correlate physicochemical properties with possible effects, and don't make mistakes with these aspects. So this is something that we do in our lab constantly, go through a lot of physicochemical characterization, both in powder and in suspension. And then we have two possibilities to study toxicology. One is using classical toxicology assessment, and this is study cytotoxicity, genotoxicity, possible uptake, possible oxidative stress. We go through this classical analysis as well in our lab, but we think that there is another way to go, let's say, through this shortcut strategy, and this is to utilize methodologies that could provide a more holistic information of what is the risk and what is the effects and the impact into the cell. And OMICS is a series of techniques such as proteomics, studying proteins in the cell, or lipidomics, study the total content of the lipids in the cell, or transcriptomics, by study the transcripts and how they change in the cell, that give this information in a more complete way. And actually we can connect it, all of them together, to get a more clear answer to what is happening. So to give an example, I put it here, how we try to understand the possible cellular effects and functional impacts by studying different types of cells in vitro cell culture. Here I put, for example, keratinocytes or endothelial cells and exposed to nanoparticles, and then try to see what has happened after this exposure and to understand what is the possible risk for the cells to be exposed to those nanoparticles. So to take a piece of example here about hepatic toxicity of nanoparticles, in this paper we have been combined two types of omics approaches. One was classical proteomics and lipidomics. And by combining that in a type of a scenario where we use nanoparticles from metal oxides with hepatocytes, we come up with a kind of conclusion that gives us a kind of degree of impact of the different nanoparticle in that context. So we have an information which one is more risk than the other in exactly the same scenario. So this is a piece of the data from that approach, and I want to point it out here only a little bit of information, and this is that the correlation between this methodology that help us to understand the question. Here we can see from the data of the proteins, for example, that we have a specific effects in metabolism of lipids at the protein level, and also we can see from the information of the distribution of the lipids and how the lipids change in the membrane, that also we have this effect. And we have changes in sphingomyelin, for example, in the small nanoparticles of titanium dioxide. So this is a way to see how omics methodology give analytic information, could be combined between different of them and get a more clear answer with one single methodology. Another possibility is using transcriptomics, and today RNA-seq analysis is one of the techniques that has become very popular and very powerful. Here we present some data using this technique, and we can also combine with previous data, and we see here, and this is the reason why I put this, that also the same type of nanoparticle from the previous study, this ultra small nanoparticle, also from this technology confirm our previous result and confirm this aspect of the lipid metabolism that were specifically involved in the impact of this nanoparticle in that cell. So all of these methods that I present in the previous slides are related with in vitro studies, and I want to finish considering that obviously in vivo studies are important, but are very complex, costly, and therefore when you want to go to the final in vivo toxicology, you need to find what is the best model for this purpose. And actually I think in nanotoxicology still one of these possible methods is utilize zebrafish embryo model, and in this faculty we have also this facility. So we have collaborated with one of our colleagues in this study, and we could confirm previous results from in vivo study with this final aspect of what's happened when the zebrafish embryo is exposed to the same nanoparticle. And in this in vivo, you can come to the organism perspective that I presented at the beginning. In the in vivo, we still are into the level of the effects into the cell. And when we come to in vivo, we can really correlate it with the effects in the organs that we see at the beginning. So I hope I could explain you how these omics methodology are a kind of shortcut as well to answer this question of how safe is nano. And my final panel is related to nano risks. And what I want to present here is this problem related to material science preparing nanoparticles with a specific purpose, preparing different formulations, and they don't know which is the most safe of these formulations to go ahead with the studies. But still they need to make risk assessment of all of them. That is costly and time consuming. What we try to offer here with this method is a kind of integrate nano impact index that by using a select number of endpoints related with toxicity assessment and select a select number of cell types, you could perform all of these calculations for different formulation of nanoparticles compared to a control and end up with a value that is sufficient to give for you to take a decision which of those formulations is the safer and which is the less safe and which are the ones to go ahead for further studies. And we performed these studies with supraparamagnetic nanoparticles with a core, and then we use different coatings that are developed for different applications, and then we end up with the information for our partners and our collaborators, so which of them could be the more safe and which is the one that maybe could go ahead with a further studies or further test. So I want to put this a little bit in a slide to show you with an example of titanium dioxide that I have presented several pieces of data, how you could combine in different techniques by screening, by compare bulk materials with nanoparticles, by correlating the physicochemical properties of the nanoparticles with the effects, by applying methodologies that are a shortcut of giving holistic information in a single methodology, like I presented with proteomics, lipidomics, or transcriptomics, actually can be correlated with in vitro studies and with in vivo studies. And finally, how it's important to develop methodologies like our integrate nano impact index that has prediction capabilities. I think I have a few more minutes. And I want to show you that my questions here were nanotoxicity and nanosafety. And I hope I could address some of these aspects of the difference between nano and bulk, and by these methodologies, and how these methodologies help us to understand how the nanoparticle that is not pristine, that is surrounded by molecules, is changes into the interaction with the cells, and how also change the cell if this is internalized and make specific effects. I would like to jump into the last question because I want to present you that these techniques that I showed you there for studying nanotoxicity actually could integrate in the platform, and this is something that we are developing now in our lab, that is a kind of nanosafety platform that could help safety by design of novel nanoparticles and novel formulation. In this platform, we can evaluate novel formulation for nanodrugs that should deliver by different routes, oral, gastric, blood, or muscle, that will interact with different body fluids, and we can evaluate this, we can mimic this biointerface, we can quantify the evolution of the nanoparticle and the surrounding components that, and how this evolved, and what we can give predictions, and prediction of uptake, prediction of targeting, and prediction of risks. And just to conclude, I will take 2 minutes, no more, to tell you that all we have been talking about today, it was related to possible impact of nanoparticles in cells, in human cells. And I still think that maybe the strong risk today of effects of nanoparticles are actually correlated with the risk of release into the environment and maybe release into other species. So this is kind of a scheme of what could be the life cycle of nanoparticles and how these consumables or nanoparticles in consumables actually could end up into our environment and could end up in water, in terrestrial soils, could end up therefore in plants or in different type of animals, and then fish or other type of animals that become consumables. So actually Understanding also the possible risks of nanoparticles in the environment are actually one important concern for scientists in order to also preserve the possible impact on humans as well. So we work on that also in our lab, but I will not discuss about this aspect. That will take another lecture. But I want just to present one single slide to present one of the techniques that we utilize in order to try to understand the possible risk of nanoparticles and other components in the environment. And what the technique that we utilize to do that is also based on analysis of proteins like proteomics, but it's a kind of a step ahead where we don't analyze only the total component of cells, the total component of proteins in a cell, like in the proteomics, but we analyze all the proteins that are in a particular environmental sample. For example, if we use soil or sediment from the sea or a water column, there is also a lot of organism there. So by using quantitative analysis of the proteins of this specific environment, we can extract the proteins from different organisms, from different communities. For example, we focus in microbial communities that are very, very relevant. And we can get the information of how this change. This information gives, in one single technique, a really broad information of levels of complexity, because we can see which proteins have been changed that belong to a specific field. So we can see which organism has been disappears in our environment. We can see how the biodiversity of this specific habitat has changed. But we can go a step forward because we also can see and we know which are those proteins. And we know which proteins are involved in specific pathways that are important for that community. So we can see changes in the equilibrium of the community that involve this specific habitat. So the goal is transform through these techniques, this information about taxonomy analysis, change in biodiversity, connected with metabolic function that also are changed, and connected finally with the community, how the microbial community respond to this stress that in this discussion of today's nanoparticles that has been released in the environment. And we would like to make prediction, we make models that help us to understand this beneficial, detrimental effect of this exposure and get conclusion of what is happening and what is the risk and what is the benefits and how the environmental change after exposure to this specific component. So we work in this direction also in our lab. And now I want to conclude our talk of today, going back to my first slide where I talk about benefits and risk, and nanosafety and nanotoxicity. And my take-home message comes to these 2 points. This century is most likely one century that we could remember as a century of nanotechnology. But in order to get all of potential of this opportunity of develop new compounds, new nanoparticles with a specific function, we really need to go to safe by design platform of analysis of this component at the very, very beginning to offer the opportunity to modify at the early step before going into production and release and risks. So this is our idea how scientists should work to get this balance to the level that we will talk of nanosafety in the future and none of nanotoxicity. So thanks a lot for your attention. Here I put names of many of my colleagues that has participated in different projects and collaborated from different universities, and in particular my team of talent scientists that work with me in this project. Thanks a lot for your attention.
Okay, thank you very much for very interesting presentations. Do you have any questions?
I was wondering if this protein coating depends only on surface chemistry, or are there, I mean, given a nanoparticle with, I don't know, X charge, will Y proteins always coat to this nanoparticle?
It's not clear. I think this study of nanoparticle corona is a quite new area. So now, it has been a started study, but not as, let's say, with such accurate information that we can obtain today. So using this quantitative proteomics approach, now we can get really the 300, 400 proteins that are there and really accurate quantification. And this was not possible before. So there is still not sufficient data in order to get that conclusions, or that general conclusion of the relationship between these physicochemical properties, this just chemistry or surface chemistry with what we obtain. It doesn't look like so far from the data that is this over there, but it's not just interactions, is maybe the combination of all the forces, obviously, and depends of the sizes and depends of the coating. But also now we start to evaluating also if there is a difference between, I mean, we need to know the dimension of this corona and how this dimension change, and if there is also difference, if homogeneous distributed or is distributed in a specific, let's say, raft or a specific package of proteins. All of these questions still are really new in this field, so are not, they haven't been answered, or at least there is data from one nanoparticle in one condition, but not sufficient data to get to kind of universal conclusion of how is happening, or what is the main driving forces. Most likely combinations.
I'm involved in the antibiotic resistant research, and I learned that there are outer membrane vesicles in, for example, Moraxella catarrhalis bacteria that could help the bacteria to get rid of antibiotic effect. Do you know anything more about that? So it should be small nanoparticles, outer membrane vesicles, in some gram-negative bacteria.
Yeah, sure. I mean, I don't know this particular topic, but of course, nanoparticles most likely can transfer this auto and then get into the intermediate area. So if they come into a kind of eukaryotic cell, definitely also they can go into prokaryotic.
So are there any options to treat or to kill these bacteria by using this mechanism?
I don't know, it's completely out of my field, but I don't know if people are really studying this and develop something. It seems likely, but of course you need to design the things in the proper way, understand also the functionality of all the elements. So it's something completely out of my field to define, I mean, to come to an idea how to do it, but it's maybe possible.
So what do you think in medicine, where will the first breakthrough be in nano?
In nanomedicine.
Nanomedicine, is it in cancer treatment?
Yeah, I think cancer, there is a lot of things going on now. Obviously, there are many steps to come to a kind of something that go to the hospitals, but it's important, so there are a lot of interest and investment on that, and it's also I think there will be different depending on which type of cancer. But with some specific cancer that maybe are very localized and easy to very accessible, it's maybe possible that some formulations were able to target, to deliver the component and to make an effect, a particular local effect and not effect in the other cells. That is the goal. So I think this is an area with a lot of research around, and most likely is the things that we will see first.
Any more questions or comments? Okay, so otherwise I will close the session, and thank you very much for coming here, and thanks a lot.